Right, so I'm Jeff Johnson. I'm working in the Spec Lab, and I'm going to start talking about post-translational modifications. I think when you talk about mass spectrometry and how the technology has advanced in the past 5 or 10 years, it's really easy to see how powerful it is when you look at post-translational modifications. There are a lot of things that I'll talk about today that you probably couldn't have done even 5 years ago. So it's really technology that opens a whole new field of research that couldn't be seen before. I probably don't have to convince anybody here that PTMs are important, but I will anyways. PTMs are absolutely critical for fast biological processes, especially for transducing some sort of signal and changing a biological state very rapidly. They're typically, all the components are already present, they just have to be activated or deactivated, often by another PTM or binding to a cofactor or something. And then once they're activated, they can very rapidly transduce the signal. These are very fast enzymatic reactions, right? But on the other hand, it presents a sort of technical challenge that these reactions are so fast, you often can't identify enzyme-substrate relationships, for example, just by looking for physical associations, which is often your first go-to for understanding a kinase or a PTM substrate. It just doesn't work that way, right? They're too fast, you can't capture those reactions. And so this is where I really think that shotgun proteomics and developments in mass technologies can allow something that you couldn't do before, which is you can look for enzyme-substrate relationships not by looking for physical associations, but by looking indirectly. If I inhibit a kinase, and I look at all phosphorylated proteins, and I look at the ones that have changed in response to that inhibition, you can actually identify PTM substrates that way. And again, that's something that you couldn't really do 5 years ago, but now technology is so fast, the methods are so reproducible, you can very easily see essentially all phosphorylation and look for just those things that are changing, right? And then of course, PTMs are frequently dysregulated in disease. And again, I don't have to prove anything to you, probably you know PTMs are important, but let's just say we know they're very important for cancer when signaling pathways run out of control. We know that pathogens love to target PTM enzymes, kinases, ubiquitinases, deubiquitinases. And then for diseases of protein folding problems, a lot of dysregulation with protein folding also is related to PTMs, especially ubiquitination. Right? So PTMs are important. In our lab, there are essentially 2 questions that we get very frequently that users come to us to ask related to PTMs. And the most frequent one is I have this protein of interest and I want to know what PTMs are regulating my protein and where on my protein are those PTMs located, right? And then the other question is I have some condition, like I have a kinase inhibitor that I know has a phenotype in my system, and I want to know what are the PTMs that change in response to that condition, right? And so this is more a global question. Like I said with the kinase inhibitor, I want to look globally at all phosphorylation to see what phosphosites change in response to that condition. And it may or may not surprise you, but often the first question can be very difficult to answer. But today, in 2017, the second question is actually, depending on the PTM, is a pretty easy question to answer. So I'll just go through both of those things. I'll talk about some strategies that we use in the lab to answer those questions. And I'll start with this first one, which is PTM mapping on a protein of interest. So this technology hasn't really advanced all that much. Essentially, we're not using any fancy tricks here. Whatever you give us, we're going to digest with an enzyme, probably trypsin, maybe 1 or 2 other ones, and we're going to inject them into the mass spec. There's not a whole lot else that we can do with that. The exception that I'll say is that there is a very specialized approach called top-down proteomics, which you're going to hear later in the workshop, where you take the intact protein with all of its PTMs on it and you put it all into the mass spectrometer and then fragment it within the mass spectrometer to localize any PTM. It's not easy, it's very protein dependent, but when it works, it's a beautiful technology. But that's not what I'm going to talk about. So when we're mapping a PTM of interest, you come to me and you say, what PTMs are on my protein? I come to you and I say, how much of it can you get? Because this is really, it's not a complicated problem here, right? We're just constrained by the limits of the mass spec. So if you figure the limit of detection of the mass spec is about 1 femtomole, actually lower than that, but that's like my comfortable range. And actually, I don't want to operate at the limit of detection. I want to operate about 10 times more than that. So say 10 femtomoles. And I don't want enough that I inject it once and it's all gone. I want at least like 4 or 5 injections of your sample. So really you're looking at, just to detect the protein, I want about 100 femtomoles to start with, right? So for a 50 kilodalton protein, that's 5 nanograms. That means you should be able to see your protein on a silver stained gel. That's what I tell everybody. If you want to see it on mass spec, if you see it by silver, you'll probably see it. So that's just to see the protein. Now we're talking about PTMs. The next question I would ask is, do you know what the stoichiometry of your PTM is? How much of your protein is actually modified compared to unmodified? And usually you don't know the answer to this question, but it's an important one to think about, right? If you're overexpressing a protein, the PTM enzyme that's going to modify it is not being overexpressed, so there's probably only a fraction of it that's actually modified. If your protein exists in multiple different locations, or it's in different complexes in the cell, and you're purifying all of those complexes, only a fraction of it is going to be modified, right? So keep that in mind. So if only 10% of your protein is phosphorylated or has a PTM, then you need 10 times more than that, so you need now 50 nanograms of protein. So now we're talking about Kumasi visible values, right? If it's less than 10%, if it's more like 1%, then you run into another problem, which is I can only load so much onto the mass spec before I overload my column, and I reach a sort of saturation on the mass spec. And so if you're less than 1% of your protein is modified, then you need to take your sample, scale it up even more, and enrich for the PTM of interest, and then just inject those in. Does that make sense? So in most cases, again, every protein is different, every PTM is different, but in most cases, if you can see a band in a Kumasi stained gel, that should be enough for us to get some PTMs, unless they're very low stoichiometry. But again, 50 nanograms is not that easy to purify for some proteins, right? Now the other question is phosphorylation, or integrating with other data types, either other proteomics data types, or some sort of functional or genetic screen, sort of narrow in on what's important. So coming to this question of the HIV-dependent phosphoproteome, so it was very much the case. I got an Excel table with many thousands of lines in it and a bunch of things that were changing, and I thought, I don't know what to do with this. So we have a collaborator, Pedro, who used to work in our lab, and now he has his own lab at EBI. And he developed this tool that was originally called PTM Funk, and now it's called Phosphate, and you can basically plug in a phosphoproteomics data set and it'll run all of these really cool analyses for you. But this was a while ago. So we basically took PTM Funk, which is a curated list from the literature, essentially every phosphoproteomics study that was out there, that was annotated, put it all into a database, and then we took our HIV-dependent phosphoproteome, so basically the full changes in p-values for the HIV data, and compared it to PTM Funk and said, what in the published literature looks like changes that are HIV dependent, right? And so what this bar graph is it was something like 45 or 50 different phosphoproteomic studies that were in that database, and we calculated the correlation of our HIV data with each of those different data sets. And what you find is that there are 2 different cell cycle studies that specifically the mitosis time point correlated very tightly with the HIV-dependent proteome. And it's actually known that we should see that because HIV arrests cells at the G2 to M phase transition. So what you're seeing there are things like CDK1 double phosphorylation to inhibit the cell cycle. But what was interesting was at the opposite end of the data, there was data on an aurora kinase A inhibitor that was anti-correlated with the HIV-dependent proteome. And so we asked the question, are there aurora kinase A dependent sites in the HIV-dependent proteome, and is HIV sensitive to aurora kinase? And to answer the first question, the answer was Yeah, there are a lot of known aurora kinase A substrates that were in our data. In terms of the motifs that we pulled out of our data, and again, this is another tool that's very good for phosphorylation. We found there were two motifs that were enriched. The first is an SQ motif, and that's again this cell cycle arrest. It's the ATM-ATR recognition motif. But the second one, this R in the -2 position, and there's actually an RRK in the -3 position also, that's consistent with aurora kinase A phosphorylation. But to really drive the point home, when you take an aurora kinase A inhibitor, and this is actually the same inhibitor that they used from the previous study, you can see that there's a very strong dose-dependent response, killing HIV in response to aurora kinase A inhibition. So that's one way of using bioinformatics to sort of focus in on functionally relevant things. One other approach is just by throwing a bunch of other conditions at it. And so here we're looking at same system, Jurkat cells that are infected or uninfected with HIV, but now we're looking at ubiquitination. And so obviously for ubiquitin, if you block the proteasome, you can stabilize things that are ubiquitinated and degraded. So we did both the infections in the presence of MG132 or in the absence of MG132, as well as doing protein abundance measurements for those things. And I'll just skip ahead to say that by looking for things where you saw an increase in ubiquitination, but a decrease in the baseline protein abundance, you can focus in on those things that look like they're being ubiquitinated and degraded. And there's really not that many things that fit that profile, and so we showed that there's a B56 subunit of PP2A, a regulatory subunit, that's degraded specifically by VIF. When you delete VIF from the virus, it's not degraded. When it's present in the virus, it gets completely degraded. And then finally, just one last example. We looked with Jeff Cox at different infections with bacteria. We looked at MTB infection, Listeria monocytogenes infection, and Salmonella enterica infections, looking at what was ubiquitinated in each of those different bug infections. And here is the case where we gave them a list of things that were changing. They looked at that list and picked the top 10 or 20 or so and overexpressed them one at a time to see what effect it had on bacterial replication. And one of those hits that was in the top 10 is a protein called OASL1. And when you overexpress that protein, it restricts MTB growth. And they were able to show that it's related to IRF7. And then what was interesting is if you look at the protein's actually not changing all that significantly. It's being degraded to some extent. But at the mRNA level, OASL1 is an interferon-stimulated gene. So the mRNA level is increasing something like 8-fold or 10-fold, but the protein is actually decreasing. So this is a sort of thing where gene expression wouldn't be able to give you this sort of information. Even looking at the protein, you wouldn't find that very interesting because it decreases by maybe 0.8-fold. But at the ubiquitination level, it was ubiquitinated at least 2 or 3-fold, and then we were able to go back and figure out what it was doing that restricted TB growth. So those are my examples. Sorry, I had to go really fast. It was a short time. But these are all the people who contributed to that work. And I'm over time, so I don't think I have time to take questions, but I'd be happy to answer questions during the reception. So we'll move on to Danielle. Alicia.